And yeah, take it away.
Okay, hello. Today I want to show you some work I did while I was in the lab of Rob Knight in San Diego. Meanwhile, I moved on to Düsseldorf, and this work is in the context of amplicon sequencing, where we have different techniques to get exact amplicon sequences or sequence variants or sub-OTUs, however you call it. You address a bacterium by its nucleotide sequence. And we want to place those nucleotide sequences into a phylogenetic tree to do the usual alpha and beta diversity computation. And we do all that to improve some associations with clinical information. So this is the motivation. We need the phylogenetic tree to compute alpha or beta diversity in a phylogenetic manner. So with Faith's PD for alpha diversity or beta diversity, there we use unifrac unweighted or weighted unifrac. And in an ideal scenario, we would be in a situation like here where the black sequences are full-length ribosomal sequences. We have a complete reference. We do our sequencing amplicon experiment. We get those, for example, V4 short sequence reads. And then we can use the reference phylogeny, for example, green genes, and use this existing tree to compute diversity. However, if we go to environments that are not that densely sampled, like ocean samples, then we often are in a situation where we have just a few full-length reference sequences for the ribosomal sequence available, but we get a lot of amplicon sequences. And then the challenge is to include those additional amplicon sequences into the phylogenetic tree. The traditional method is using a closed ref approach, where we have the green genes tree, for example, or silver or other trees. And we use some clustering approach like SORT-me RNA, where we take the sequence variants and want to assign them to already known tips in the reference. This works for some environments, for some others it doesn't work that good, and we lose a lot of newly observed sequence variants. Alternatively, we could compute a de novo tree only out of those small sequence variants, but we don't have enough phylogenetic information in those short, let's say 150 nucleotide sequences, so we don't know how well the quality of this phylogeny is really. And therefore, I want to here present you the approach of an insertion tree, where we combine both methods. So we start with this reference phylogeny. This is the black backbone tree here. And then we want to take our newly observed sequence amplicons and insert them, hopefully at the right position, into the reference tree. That's the overall approach. And then we have a new phylogeny. I did a lot of evaluation, and here are some warnings at first. So this is a warning that you might not want to use a de novo tree computed only from your short amplicon sequences. So this here is an example where we had an actual experiment with 600 samples, and we deblurred our sequences to get exact amplicon sequences, and we constructed a de novo phylogeny, computed unifrac, and did this PCOA dimensionality reduction to get to this picture. And what we see is a huge separation of those samples, which couldn't be explained by any metadata variable. So I was looking into the tree, and I found that there's one tiny little clade that is very far outside of the rest of the tree. So it seems to be some computational artifact. In fact, it was a clade with only 3 sequence variants from this Methanobrevibacter. And here I'm coloring my samples by the abundance of those 3 sequences. And here are very few, and here are none. So this rarely abundant taxon drove the separation of samples. I think it's an artifact because of this long branch that sticked out. I manually shortened this branch, and I did the recomputation of unifrac, and I got this result here, so I could resolve the artificial clustering. Or I'm using this insertion tree automatically, And I also get the artificial clustering resolved without any manual intervention. So I recommend not to use de novo trees at all for your short sequence amplicon reads. Why do we want to use it? We think that we get a higher resolution for subtle differences. So if we combine it, or if we use the combination of some exact amplicon or denoising strategy like ampliclust, which we heard about yesterday, or DADA2, or in our case dblur, and the insertion tree, then we might be able to improve the statistical power to find differences in microbiome samples. So here the traditional approach would be using closed reference, and we have here 109 samples of children in Malawi with good gut health, however good is defined, and poor gut health. And in this classical approach, we are not able to have a sufficient p-value if we run a Permanova test. The p-value becomes better. I'm not sure if you can read it. It's here 0.04. If we run an open reference approach, which is somewhere in the middle between a closed reference and a de novo approach. But here with the method I'm presenting, in this case, we have enough statistical power to really tell apart the different samples from good and poor gut health, gut metagenomes. What we also can do is meta-analysis, even in cases where we sequence different variable regions from the 16S gene. So it's completely impossible if we want to do it in a combined fashion with, in this case, V4 and V2 for two experiments which target the same body sites. You see this artificial line up here of all the samples from the one experiment. The other experiment is fairly well. Then we use the insertion tree idea to insert the sequence reads from V4 and V2 regions. And as you can see, the clouds merge by body site, which is good. You still have some separation between the one experiment with the spheres and the other experiment with the cones. But I'm not sure if this is biologically driven or not, because these are very different populations, Westernized and Indian people. And in this lucky case, as a control, we had also V2 reads from both experiments, which is positive control, so it gives you the right picture. And I hope it's very easy to use, so I spent some time to wrap all the software up. And once you have a CHIME environment in your machine, you can easily install this plugin as a community plugin. So all you need to do is say conda install Q2 fragment insertion. This is the name of the plugin, which gives you the insertion trees if you want to compute on your own data. Or alternatively, if you're already using our Cheetah framework where you upload your samples, it gets automatically computed, and you get here one button where you can directly click and download the insertion tree. With that, I want to thank my team in San Diego, and I'm open for your questions.
I think we might have time for one quick question while we change speakers over, if there is a question. I have lots of questions, but I can ask them later.
Yeah.
Okay, can the next speaker please come up?